Hello everyone, my name is Damiano Sgarbossa and I represent a joint work that I did with Umberto Lupo and Florence Bitwell. In this work, we propose to use mass language modeling to correctly infer protein interaction partners among paralogs. So let's start with a brief introduction on proteins. A family is a group of protein sequences called homologs, which evolved together from the same ancestor, and they have a similar structure and function. We can describe families using multiple sequence alignments, which are simply a collection of protein sequences from the same family, which are aligned with each other. And to make it simple, we can just make an analogy between protein sequences and natural language. The basic unit in natural language is the fundamental unit in natural language are words, which in our case, the fundamental units instead are the amino acids or the residues, which are represented by letters in this picture here. Then if you join words together, you make sentences, and in our case, joining amino acids together, you get protein sequences. And finally, if you collect together multiple sentences, you can make a document, while in our case, we have multiple sequence alignments, or MSAs, which are made of aligned protein sequences, while instead in documents, the main difference is that in documents instead, the sentences are typically not aligned with each other. So, since we can train large language models on natural language, we can do the same for protein sequences. In fact, the model that we used is called MSA transformer, and it's a BERT-like transformer model which was trained to correctly predict randomly masked amino acids in single family MSAs, which is what you can see here in the image. And the model masked language modeling objective, masked language modeling loss, is simply a cross entropy loss on the masked residues or amino acids. So, in summary, the problem that we want to solve is the following. If you have two protein families, A and B, for which you have two alignments, two MSAs, you want to match each sequence from one protein family with its correct interaction partner from the other protein family, which is protein family B. And in general, protein sequences coming from the same family and the same species are called paralogs. And you have several paralogs for species, as you can see here. And therefore, there are like the number of possible one-to-one matchings between sequences in one family and the other scales factorially with the species size. And equivalently, we can describe this as constructing a concatenated multiple sequence alignment in which we concatenate family A and B together, and such that in the same row you have the correct interaction partners. And the usefulness of all of these is that if you have an MSA like this, you can use it for protein structure prediction of complexes, which is quite a hard problem right now. So we proposed a new approach to solve this matching problem, which is based on using a pre-trained language model, which is MSA transformer, and its mass language modeling objective. So here, even though this model was trained on a single family MSAs, we feed to the model a concatenated MSA made of interacting families, which are in this case, like before, A and B, and we compute the mass language modeling loss. The idea is that the model will use information from protein family B to predict the amino acids, the masked amino acids in protein family A, and vice versa. So what we hope is that the model will perform better on correctly concatenated alignments with respect to wrongly concatenated ones, giving a lower mass language modeling loss. And indeed, this is what it does, because it performs better on correctly concatenated MSAs. As you can see here, in fact, we have that the mass language modeling loss decreases for increasing number of correctly paired sequences. And this suggests us a solution to solve this problem, which is simply matching the paralogs by finding the matchings that minimize the masked language modeling loss. But still, now we have the problem of the factorial number of possible permutations between all the sequences. And to solve this, we use a method presented in this paper in iClear 2018, which formulate a differentiable optimization problem that can be solved efficiently using gradient methods. And therefore, what we do is like looking for optimal permutations by smoothly parameterizing the nearby soft permutations. So in conclusion, we tested our methods with some data sets for which we have the ground truth. We saw that in the regime of shallow MSAs, which means that not many sequences, we're able to consistently outperform the coevolution-based state-of-the-art methods. And we also observed that adding some fixed context, which means sequences for which we have the ground truth, improves monotonically the performance of our method. Thank you very much, and also I would like to thank my collaborators and my entire lab.